Hello everyone, in this video we're going to talk about the central dogma, transcription and translation. So transcription is DNA, using the DNA code to make RNA, and then RNA is then translated into proteins to actually function in the cell. So you can kind of think of it as DNA is kind of like the computer code that is fundamentally what runs everything, but it really doesn't, it isn't very practical. But then proteins is what we call the expression of the gene. It's what actually gets the job done. So before we dive into transcription and translation, we want to talk about three different types of RNAs. So as you may or may not already know, there's one type of RNA called the messenger RNA. So the messenger RNA is very important, actually. All of them are important, but messenger RNA is the only type of mRNA that's actually translated into proteins, right? Sending a message from the DNA into protein. So we're figuring out how to get this computer code to run. So mRNA is kind of a step to run the code. So mRNA in a prokaryotic cell is transcribed and translated in the cytoplasm. But in a eukaryotic cell, mRNA is kind of a messenger boy that sends the message out of the nucleus, because DNA cannot leave the nucleus and mRNA can. And then mRNA is then used as information or instruction on how to construct the proteins. So if you look at this picture right here, we have this pink line, and that signifies the mRNA that's getting translated at this large molecule, which is the large organelle, which is called the ribosome. The next one that we want to talk about is called tRNA. tRNA is called transfer RNA, and transfer RNA is good to transfer amino acids. So during translation, individual amino acids are connected together to form a polypeptide chain. The reason why it's called a polypeptide chain is because when amino acids are connected together, the kind of bond that allows them to be attached together is called a peptide bond. It's a type of covalent bond. So tRNA is used to transfer specific amino acids. So when we say specific, what we mean by that is there are different types of amino acid and there are different types of tRNA that can only carry one type of amino acid. So each type of tRNA is directly associated with one specific type of amino acid. And then where does the tRNA go? It goes to the ribosome. So again, when you look at this picture, you see the mRNA right here, and then you see the tRNA. What you can tell is that on the mRNA, there's something called a codon, which are three bases that the ribosome can read. And then we have the tRNA, which has three bases that can match with the codon. So these three bases are called the anticodon because it's opposite of the codon. And then we have the amino acid that's supposed to be transferred onto this growing polypeptide chain. Lastly, we have something called the rRNA. rRNA is called the ribosomal RNA, and it's a part of the ribosome. So ribosome, although it's used to make proteins and it reads mRNA, a ribosome itself is actually made out of protein and RNA at the same time. And the type of RNA that's functioning inside a ribosome is called the ribosomal RNA. Moving on. So when you look at this picture, you can see transcription is using a DNA code to transcribe that information into an RNA transcript. So you don't ever want to say DNA is made into RNA. That doesn't work because DNA and RNA are two complete different molecules. Although they're both nucleic acids, they are different molecules. So DNA being transcribed into RNA is fine. Or we're making RNA. RNA is made using the information or using the code from DNA. That is also fine. You want to avoid the urge of saying DNA is made into RNA. And then the next step is called translation. So translation is when RNA is used as the direction on how to assemble your polypeptide chain. So you, again, want to resist the urge of saying RNA is made into protein because they're two complete different things. You can't make RNA and magically turn it into protein, okay? So transcription and translation. So in this picture, I want you to take a look at the differences between prokaryotic transcription translation and eukaryotic transcription translation. So the first thing that you want to notice, or first thing that you might notice, is that in the bacteria or any type of prokaryote, we don't have a nucleus, right? But a eukaryote does have a nucleus. Something else that you can see is fundamentally the DNA in the eukaryotic cells are linear shaped and bacteria has circular DNA. The next step, so if you're moving on to transcription, you can see that transcription happens inside the nucleus for a eukaryotic cell. And for a bacteria, transcription simply happens in the cytosol or the cytoplasm. And what you also see is that before the RNA can travel out of the nucleus for a eukaryotic cell, it has to be processed. So we have to process the mRNA before it gets sent out of the cell where it gets attached to the ribosome and allow translation to happen. However, in bacteria, because we don't have a nucleus, transcription and translation can happen at the same time inside the cytoplasm. So let's talk about the first step. The first step is called the transcription. So if you look at this picture right here, you can see DNA winding together. That's the two blue lines. At the site of transcription, this DNA has to unwind first, right? Just like DNA replication, as we talked about before, we have to unwind the DNA first to allow actions to happen. And once the DNA is unwinded, then RNA bases, AUCNG instead of ATCNG, AUCNG bases can come in and match with the bases on the DNA. So form a complementary base pair of DNA and RNA, kind of DNA-RNA hybrid. And once that job is done, you have an mRNA being made. So this process, transcription, is DNA being transcribed into mRNA, as we already said, and this happens inside the nucleus only if we're talking about a eukaryotic cell, right? There is no nucleus in the prokaryotic cell. So DNA, again, can never leave the nucleus. There are a lot of words over here, but the key words is, as I already said, DNA has to be unzipped first, and then mRNA is assembled by pairing the complementary bases, right? So if you look at this picture, you can see that A is matching with U, T is matching with A, C is matching with G, G is matching with C. So anytime you have A, you match it with U instead of T, but when you're given a T on the DNA strand, that's not a problem because there's always A for an RNA. So remember, RNA uses U instead of T. There is no T base. That doesn't exist at all for RNA nucleotides. And remember that RNA is often single-stranded. It's not always like that, but for our purpose, mRNA at least is single-stranded, okay? So moving on to the next page. On this picture, you can see the 5 to 3 and 3 to 5 direction of the DNA, again, anti-parallel DNA, double-stranded DNA, right? So what you can also tell is that the RNA, the mRNA that we're producing, is 5 to 3 this way, and then DNA is 3 to 5 this way. So even when we're matching RNA bases onto DNA, the directions are still opposite. We still have anti-parallel RNA versus DNA. Something else that you can notice is that there's a coding strand and a template strand. So the template strand of DNA is used as a template. So the template strand is what we use to actually add the RNA bases onto. So the RNA is complementary to the template strand of the DNA, right? But DNA has 2 strands, so the other strand is called the coding strand. The reason why it's called the coding strand is because it technically has almost the exact same code as the RNA that's being made, right? So as you can see, this T on the template strand matches with an A on the RNA, and it matches with an A on the DNA, right? This A is an RNA nucleotide, and this A is a DNA nucleotide. They're not exactly the same, but they have the same code. The only thing that's different between the template strand, or between the mRNA being produced and the coding strand, is the U and the T. Everything else is exactly the same. As you can see, G and G, A and A, C and C over here, and we can keep going, right? Moving on. So if we're talking about a eukaryotic cell, a lot of the actions that take place in the eukaryotic cell is simply more complex, So one of the things that happen in a eukaryotic cell that doesn't take place in a prokaryotic cell is something called RNA processing, or post-transcriptional modification. So what we mean by that is that once we have the mRNA transcript made, this mRNA has to be processed in the nucleus before it can go out of the nucleus. So before we look at the notes, you can take a look at this picture right here. So on the DNA strand, you see exons and introns and exons and introns, and you keep going. So there are segments on the DNA called exons and introns. Similarly, when we transcribe this piece of DNA into RNA, it also has exons and introns. So here's the thing about exons and introns. Introns are kind of gaps in between exons that needs to be taken out. It's almost like you have a piece of paper, you print a star on it, and you want to put the star on your wall for some reason, and you have to cut out the star, and then you have to throw away the rest of the paper into a recycle bin, and you recycle it. Same idea with this. You have RNA, you have the introns, you have to take the introns out, and then repiece the exons together to actually form a mature mRNA. So this mRNA right here is the mRNA that's actually used as a code to make protein, or as the instruction to make protein. If you were to make this entire piece the instruction to make protein, this protein is not going to be right, okay? And later we'll talk about why introns exist in the first place. So again, introns are the ones that does not code for proteins, and it interrupts the sequence of genes. And then exons are the coding sections of the DNA or RNA, and when you group the exon pieces together, that's how you can actually allow translation to happen next. So this entire process, we give it a name called RNA splicing, because we have the RNA, we're splicing it into pieces, and we remove the introns, and we reattach the exons. So this RNA splicing is only one type of RNA processing, right? This is one step that needs to be done before RNA is sent out of the nucleus. Something else that needs to happen, this is just a dumb joke. Okay, moving on. Something else that has to happen before mRNA is ready to be sent out of the nucleus is called the 5' cap and the poly A tail. Okay, so what you see right here, we have an mRNA transcript just made, fresh out of the oven. What do we do with it? We cut out the introns and we group the exons together. However, something you have to remember is that the cytoplasm is kind of a dangerous place for nucleic acid. There are a lot of digestive enzymes that can easily break apart the mRNA, or any type of nucleic acid, really. This is kind of why DNA has to stay in the nucleus, because you don't want your DNA being destroyed, because DNA is the genetic information that's really, really important for every single cell, right? Obviously, everything else is important too, but DNA is fundamentally what directs how everything is done in the cell. So once this RNA is made, you don't want it to be sent out of the cell just to be destroyed before its mission is done. And the mission of an RNA is to be used as the instruction to make protein, right? So we got to protect our vulnerable little RNA. What do? We give it a hat, we give it some boots, and then we send it out. So the hat is kind of, I call it the hat, it's the 5' cap. So again, just like DNA, RNA also has the 5' end and the 3' end. Those are two directions of the RNA. So on the 5' end, we add a cap called the 5' cap. And then we have on the other side, the 3' end, we have the poly A tail, which is just a bunch of adenines, RNA-based, adenine, the adenine base, being added, a bunch of A's at the very end of the 3' end, okay? So we have the 5' cap, we have the poly A tail, we did our RNA splicing, now our RNA is ready to go, okay? Ready to go, okay. So the next one, earlier you might be thinking, why is it that introns exist in the first place, right? What's the point of having these interrupting sequences in the DNA and the RNA just for it to be cut out? Here is one important reason. There are many other reasons, and we really, as scientists, don't really know all the reasons yet, but this is one really important reason. So if you think about it, you have a lot of cells in your body, right? Countless numbers of cells. And every single day you have cells dying, you have new cells being made, you have cells doing cell division, you have cells in the process of doing cell division, all of that. But in the end, most of your cells, and at least all your somatic cells, should have all the same DNA, right? Because we all came from our original one cell that we came from, all our body cells came from that one cell, right? So all our body cells, all our somatic cells have the same exact DNA. But what makes your brain cell different from the cells that are in your heart and the cells that are, the lining of your lungs, whatever, right? What makes those cells different? How can that happen if the instruction in the DNA is all the same for all the cells? So here is something called alternative splicing. So alternative splicing is a way that we can regroup different exons together to produce different proteins from the same original mRNA transcript. So what you see over here, this piece of DNA has exon 1, 2, 3, 4, 5. And then as a result, the transcription allows exon 1, 2, 3, 4, 5 to be formed, and the introns as well, on the original RNA transcript. Depending on what kind of cell, this cell is supposed to be, right? That's the fate of the cell. We can alternatively group together different pieces of the exon. So one way you can group 1, 2, 3, 4, 5 together, and that will give you one specific type of protein that only exists in one type of cell. Alternatively, you can group only 1, 2, 4, and 5 together, and that gives you a different protein that exists in another type of cell. So that allows the second cell to be different from the first cell, right? Similarly, you can do 1, 2, 3, and 5, and that gives you a different type of protein for a different type of cell. So although, again, we have the same RNA being produced originally from the same DNA, because every single cell has the same DNA, you can produce different types of proteins specifically for a certain cell to allow that cell to be different. Okay, so that's called alternative splicing. After transcription and RNA processing, again, now is the time for mRNA to leave the nucleus for a eukaryote that we're talking about, okay? Where's it going? It's going to a ribosome. So this ribosome could be a ribosome that's attached to a rough ER, or it could be a ribosome that just floats around in the cytoplasm. The next page, oops. All right, translation happens at the ribosome, right? And again, ribosomes are made out of rRNA and proteins. Once the mRNA arrives, it's read in a group of 3 bases called a codon. That is how the ribosome functions. That is how the information of the mRNA is interpreted. So 3 bases at a time being read at the ribosome. Okay, so every time you look at a codon or a codon chart, what you're reading is the mRNA bases. Okay, so if you look on the right side, we have codon 1 through 7, which means every single one of these codons, unless there's supposedly there's no stop codon in there, every single one of these codons will code for 1 amino acid, and altogether they will produce 7 different amino acids, and together you have a very small polypeptide chain containing 7 amino acids as a result of translation. So a codon codes for 1 amino acid. All right, or if a question can ask you, if you have 3 codons, how many RNA bases would that be? First off, you have to know that it's RNA, it's not DNA. Codon is always talking about RNA and specifically mRNA, messenger RNA. So 3 codons will give you 9 RNA bases. The next one is when you look at an amino acid chart. There was a video specifically talking about how to read a codon chart, but basically the idea is every single codon is made out of 3 bases. You look down first to find your first base, you look across to find your second base, and then you find your specific codon that you're looking for in one of these boxes. For example, if you're looking for CAA, you look down, you find C, you look across, you find the second letter A, and then you find CAA right here. So the CAA codon, mRNA codon, is coding for an amino acid called glutamine, okay? So when you're looking at this codon chart, which will always be provided to you, there's some information that you can tell. There are 20 different amino acids that are used in the human body. That's one thing. The second thing is there are multiple different codons coding for the same amino acid. So there's way more than 20 different codons, but you only have 20 different amino acids. And there's a reason for that, okay? The reason, or for this is called redundancy. This is redundant. The reason for this redundancy is that it minimizes harmful effect that mutations can cause during protein synthesis. So think about this, right? If you have an RNA chain, or you have a DNA that codes for this RNA, and there's a mutation in the DNA that switches a base in the DNA, which then, of course, it would switch a base in the RNA, right? So let's say the switch of base, originally, you should have CUU coding for leucine, right? If it just so happened that the switching of this one base, this one mutation, changed the last base of this one codon, you change from CUU to CUA, you have a mutation in DNA. It's changing something. You have a mutation in the RNA, it's also changing something, but the amino acid is not changed. If your amino acid is not changed, that means the protein being made in the end is going to be exactly the same as the one before, which means this protein will function just fine, right? So this mutation has no effect. This is actually called a silent mutation, which we'll talk about a little more in the future, okay? So that's redundancy. You need to understand why or what's a benefit for having redundancy in the first place. So this is just to tell you that different amino acids have different properties. So if you look right here, some are, some are alkaline, some are acidic, some are hydrophobic, hydrophilic, some are basic, some are, wait, I already said that. Some are positive charged, some are negatively charged, whatever. So the idea is that how a protein is folded, which ultimately determines how a protein can function, is determined by the amino acid sequence. So you have these amino acids being assembled together in a certain sequence, right? Just like computer codes, you put all these random words together, and now it's supposed to mean something, right? You have this amino acid sequence that is determined by the mRNA, which was determined by the DNA, and then it can fold into a shape, right? Think about it. If you have a positive right here, negative right here, they can match. If you have a hydrophobic amino acid, it's going to go inside the protein. If you have a hydrophilic amino acid, it's going to want to be on the outside of the protein, right? So the shape of the protein being determined by the amino acid sequence is what allows the protein to function. So practice. DNA strand right here. How do you get an RNA? You do A to U, and then G, whatever. Okay, so you can take a look. You're welcome to pause and see if you can practice this on your own. And then you can figure out, pause again, see if you can figure out what the amino acid sequence is supposed to be as well. So that's the answer. I hope the answer is right. Okay, moving on. Almost done. So now we can talk about translation. We've done all this work. We're finally there, right? Translation is what allows... Why isn't this moving? Oh my god. Anyway, this picture is supposed to move. You have a tRNA coming in to this ribosome, 3 bases being read at a time. This is codon, this is anticodon, and we have amino acids being carried and being transferred, and they're being attached together, okay? So how is translation done? We have tRNA translates the RNA into a protein at the ribosome. Again, if you look at this picture, we have codon, we have anticodon. When they match, the amino acid gets dropped off. Okay, so it's like dropping off, I don't know, your dog at your doggy daycare because you don't have kids. I also don't have kids. Anyway, you're dropping off your dog at the doggy daycare. You're not going to drop your dog off at the grocery store. That just doesn't work, right? So same thing with the tRNA. It's only going to drop off its amino acid when the anticodon and the codon can actually match. So again, on this picture, you can see anticodon being matched with a codon, and then we have the amino acid. The next one that's coming in, and we have anticodon matched with okay. So tRNA have anticodon that is complementary to the mRNA codon. I'm going to say this one more time. Whenever you're reading the codon chart, the large chart that has all the letters and all the amino acid abbreviations, you are reading the mRNA right here, okay? The original mRNA transcript. You're never going to turn the mRNA, or in our class at least, you're never going to turn the mRNA codon into anticodon and then figure out what the amino acid is. That's not how it works, okay? When we look at translation, we're getting stuck on this video again. Okay, tRNA carries 3 bases, anticodon and amino acid. The correct tRNA molecule with an anticodon complementary to the codon being read will bind to the mRNA, right? So this is kind of all random. In the cell, you have things bumping into each other all the time. It's like your bumper card. When the right things bump into each other, whether it's 2 enzymes, whether it's mRNA and tRNA and ribosome, when they bump into each other, great things happen, okay? So when mRNA, codon, and the tRNA just so happen that this tRNA carrying the correct anticodon and the correct amino acid, it just comes out of nowhere and bump into this codon. When they match, they actually, this tRNA can actually drop off its amino acid because that's the right, the next amino acid, the next big thing, okay? An amino acid is then transferred to the growing polypeptide to form the correct amino acid sequence, as we said. A sequence of amino acid, again, is called a polypeptide, and once this polypeptide chain folds into its right shape, now it's a functional protein and can do whatever the job is, okay? The sequence of the amino acid determines what the protein is made, right? And the sequence of amino acid was determined by the mRNA sequence. The folding of a polypeptide chain influences the properties of a protein, right? The shape of this amino acid, the shape of this polypeptide chain after it's folded. That's what gives you a functional protein, okay? A protein is also modified by other enzymes. So once the protein is made, right, this polypeptide chain is made, two things that has to happen that you need to know of. First off, this polypeptide chain, which is just a chain, it's like a necklace. It has to fold. And then once it's folded, it might also need to be modified a little bit, give it a little tag to tell this protein where to go, or other things, okay? So when you look at this picture, you can use it as a practice. The next base is CAC. If you were to look at the codon chart, you should be able to tell that the next amino acid that should be added is called histidine. And here, again, a picture of translation. Here are a bunch of questions. Central dogma summary, transcription, you have DNA, you make mRNA transcript, and then you have post-transcriptional modification, the RNA splicing, 5' cap, poly A tail, and then you have translation. You use the universal genetic code to figure out what the amino acid sequence is until you get to the stop code. Oh, something we forgot to mention. Oops. You have the start codon and the stop codon. The start codon is always coded for methionine, which is an actual amino acid. So you want to write down methionine. And then once you get to the stop codon, it's a hard stop. You don't write the word stop even. You hard stop. Whatever is after a stop, just pretend it doesn't exist, okay? So I think that's all we have for today. We're talking about the central dogma. So here is my dog taking a nap, not learning biology. I like you doing a great job learning biology.